Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear students, we are in a continuation of the chapter that is molecular basis of inheritance. And today, in previous lecture, we have completed already about the transcription and replication. Today, we are going to understand first the what is genetic code. So, dear students, if we define a genetic code or we talk about the genetic code, you know that during both replication and transcription, we have discussed that a nucleic acid is copied to form another nucleic acid. Means in replication, DNA is a nucleic acid and it is going to form a DNA itself. But in transcription, DNA is a nucleic acid which is going to form another nucleic acid, that is mRNA. Hence, these processes are easy to conceptualize on the basis of complementarity. They can be easily conceptualized that one strand acts as a template and another one will have a what? A complementary nucleotides. So this is acting based on the complementarity. The process of translation requires a transfer of genetic information from a polymer of nucleotide to a polymer of amino acid. In translation, one of the important event that is needed, that is what? A transfer of information which is present in RNA in the form of ribonucleotides. And we have to translate this information in the form of amino acid, polymer of amino acid. So it is somewhat difficult. And we know that DNA or RNA carries all in genetic information, and it is expressed in the form of proteins which are made up of 20 different type of amino acid. And in proteins also there are 20 amino acid are of 20 different types. Now the information about the number and sequence of this amino acid forming a protein is present in DNA and is passed on to the mRNA during transcription. Thus, genetic code is interrelationship between nucleotide sequence of DNA or mRNA and amino acid sequence in a polypeptide. Means the sequence in the DNA will determine the sequence in the RNA, and this sequence in the RNA which is going to determine the sequence of the protein or a polypeptide. So it is the mRNA sequence containing coded information for one amino acid, and it consists of three nucleotides. So if this is the DNA, this is the particular segment, okay, from which the mRNA is formed, okay, and this is the RNA which having a particularly, see, this is the DNA, okay, actually ATGCTA and GGC is there, so thymine cannot be there. So this is the what? A particularly DNA. Now on the basis of this, if RNA gets synthesized, the RNA will have this is the codon on the DNA. Okay. Now RNA will have what? A UAC, and here GAU, and complementary to this sequence on the DNA. This is DNA. See, on DNA GGC is there, so it should be CCG. So this UAC will form one codon, GAU will form another codon, and CCG will form another codon on the RNA. So we are making it as a DNA, and here we are writing the sequence of RNA. Now this sequence of three nucleotide will be termed as triplet codon, and this triplet codon for a one amino acid. Okay, so another triplet codon will form another amino acid, and another triplet will code for the another amino acid. So here triplet codons means a codon which is made up of three nucleotide is responsible for the amino acids. Okay, and such a way the amino acid are formed based on the sequence on the RNA, which is described by the sequence on the DNA. Neither does any complementarity exist between the nucleotide and amino acid, nor could be drawn theoretically. There is no complementarity between the amino acid and the nucleotides on the RNA. There existed ample evidences to support the notation that changes in nucleic acid or a genetic material was responsible for a change in amino acid in protein. And there are so many such information. There is a direct no relationship between the DNA and amino acid sequence. But if any mutation occur in the DNA sequence, there is a change in amino acid in a protein. And this led to the proposition of a genetic code that could direct the sequence of amino acid during synthesis of protein. So, due to this thing, the particularly it was thought that might be there is a genetic code in the DNA which is coding for the amino acid in the protein. If determining the biochemical nature of genetic material and the sequence structure of DNA was very exciting, the proposition and deciphering of the genetic code were most challenging. Okay, so we know that determining biochemical nature of genetic material, that is the DNA, was very exciting, and Watson and Crick did it. But the proposition and deciphering or to derive the genetic code was very most challenging. So, in a very true sense, it required involvement of scientists from several disciplines. So, for this most challenging task, if we talk, suppose this is the DNA, then RNA polymerase will carry out to form RNA, and ribosome by a translation will form a protein. So, this to decipher a genetic code was the most challenging one. So, in a true sense, it required an involvement of scientists from the several disciplines, that can be physicists, organic chemists, biochemists, and geneticists. Such different scientists was involved in this particular one because it is a tough task. But it is initially was a George Gamow, a physicist, who argued that since there are only four bases, and if they have to code for 20 amino acids, the code should be of a combination of bases. Okay, because only four bases. Four bases is not possible to code for the 20 different amino acids. So Gamow told that there should be a presence of a combination of bases, and these are the particular different amino acid which are given on this particular flowchart. Okay, here you can see these are the different amino acid and their structure, and these are the four nitrogenous bases. This four nitrogenous bases, if we code, they can code maximum four amino acids. That is not able to code for this 20 amino acid. So, in continuation of the gamos, it is known that George Gamow is the scientist who coined the term genetic code. Who coined the term genetic code and argued that since there are only four bases, they have a code for 20 amino acid. So if we talk about the four nitrogenous bases or four bases, they can maximum code for the four different amino acid. Code for four different amino acid. Then 16 out of 20 remain uncoded. So it is not easy to say this is a correct proposition. But if we consider genetic code of two nitrogenous bases, then if we talk, the four bases are possible. Suppose AUGC and here AUGC. Now see, if we talk a combination of AA, it is combination going to be AA. Then it is going to be AU, AG, AC, UA, UU, UG, UC. Right? How it is happening? A is pairing with all four. U is pairing with all four. Then G. Okay. So it is a first position. And second position is explained by this basis. So GA, GU, GG, GC, and CA, CU, CG, and CC. So here total 16 codon are formed if the genetic code is of a two nitrogenous bases. So here still 4 amino acid remain uncoded. So if genetic code is of three nitrogenous bases or three nucleotides, if genetic code if genetic code is made up of three nucleotide or nitrogenous bases, then 4 is to 3, so 4 into 4, 4 into 4. If you do like this, then 4 4s are 16 4s are 64 combination of each codon made up have a 3 nucleotides, and such combination 64 combination are codon formed. So 64 codon code for 20 amino acid. It is somewhat correct because there is possibility that several codon are for coding for the one amino acid. It is possible. So he suggested that in order to code for a 20 amino acid, the code should be made up of three nucleotides. Okay, this was very bold proposition. Okay, so who suggested? Gamo suggested that Gamo suggested that in order to code for 20 amino acid, the code should be made up of three nucleotide, and this was very bold proposition by Gamo because permutation combination of four bases of three nucleotide in a three nucleotide, then 4 into 4 into 4 4 is to 3 it will be 64 codon, generating many more codon than the required. So this was done initially by the George Gamo, and the chemical method later on developed by Hargobind Khorana, and he was instrumental in synthesizing the RNA molecule with a defined combination of bases. There can be homopolymers such as UUU, AAA, CCC, GGG. Of single nucleotide, a homopolymer can be formed, and comolymer of a combination of two bases. So, this chemical investigation done by the Hargobind Khorana. And in Hargobind Khorana, when he synthesized homopolymers, RNA, he synthesized the UUU code for the phenylalanine, A GGG code for glycine, and CCC code for chlorine. So the question is how they did. So let's discuss that particular thing in detail. So it was not only Hargobind Khorana. Okay. So the important discovery was a result of experiments of Marshall W. Nirenberg and J. Heinrich J. Heinrich Mathey and later by H.G. Khoranov. Okay. Now, in this experiment, it is known that Nirenberg and Mathey used a used synthetic or artificially synthesized polyurna. So in this, in test tube they taken a polyurna like this, and when he deciphered the codon, he found that it is coding for a chain of phenylalanine. So if we give only UUU, and if we give our ribosome, and only a phenylalanine chain is synthesized in the polypeptide or protein, then obviously UUUU code for the phenylalanine. Similarly, if I could test tube and if I give an enzyme and the CCCC or cytosine only, it will form a poly-C RNA, and this poly-C RNA will code for a something, and obviously we know that it is coding for a proline. Similarly, if we give in a test tube adenine residues only and we give an enzyme, it is going to code for a particular polymer of a lysine amino acid, then obviously AAA code for the lysine. Similarly, if we take a test tube, if we provide a guanine residues and if we provide an enzyme, the GGGGG polymer will going to synthesize a glycine polymer in amino acid, means GGG code for the glycine. Such a way, all particularly four particular bases have been derived for the amino acid which is coded by the homopolymer. Okay, and then they have formed prepared copolymer like copolymer like Cu, 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 and then he deciphered the codon, then CuC code for the leucine and UCU code for the serine. So CuC code for the leucine and UCU code for the leucine. So like this, the whole particularly copolymer is synthesized. So by the sequence comparing the sequence, it is informed that CuC code for the leucine amino acid, whereas UCU code for the serine amino acid. Like this, several combination has been prepared, and by this several combination, the amino acid which is coded for the particular amino acid was discovered. Okay, now we have to go further. So in this, if we talk, then after this forming a copolymer, it was the Severe-Ochoa enzyme. Severe-Ochoa enzyme. It is a polynucleotide phosphorylase. It is a polynucleotide phosphorylase, and it was also helpful in polymerizing RNA with defined sequence. If we want to make a particular RNA of a defined sequence, it can be prepared by using this enzyme and in a template independent manner. Template independent method, that is, there is no need of template, only just there is a need of enzyme. So enzymatic synthesis of RNA means we need a just ribonucleotides like AUGC and enzyme that will synthesize the polymer of that ribonucleotide and the RNA will be prepared. And finally, a checkerboard of genetic code was prepared. Okay, so this is the enzyme which was used for the synthesis of polymer. So if RNA polymer we can synthesize, we can identify the amino acid which is coded by this particular one. And in continuation of that, the particular final checkerboard was prepared, and in which in this it is written that which codon code for the which particular amino acid. And if we talk about this one checkerboard, you can see this here they are showing a first letter means UCAG. So in these four boxes, you will see that the first letter is going to be U. In this boxes or in this row, you will see that cytosine is the first letter. Here A is the first letter and here G is the first letter. Same way on the second position, you will see U in this column, C in this column, A in the third column, and G in the fourth column in the second letter. And the third letter will be UCAG. So in the first row, in this first row U will be there in the third position. In second row C will be there. On third row, particular A will be there in the third position, and in the fourth one G will be there. Similarly, in all four bases will occur. So you can see here U is on the first position, and U is on the second position, and UC is on AG on the third position. So this two code on UUU and UUGC code for the phenylalanine, whereas UUA and UUG code for the leucine. Similarly, first letter is U and second letter is C, third letter is UCAG. All these four codon, that is UCU, UCC, UCA, UCG code for the serine. Similarly, first letter is a U, second letter is A, third letter is UCAG, and this first two codon code for the tyrosine. UAA and UAG code for the stop codon. Same way, in this box, you will see that U is the first letter, G is the second letter, and the UCAG on the third letter. First two coding for the cysteine, and the UGA is the stop codon, and UGG code for the tryptophan. So here you can see these three are the stop codon. Means stop codon means a codon in a mRNA where the proteins will stop. Same way, C on the first position, U on the first position, UCAG on the third position. This all four code for the leucine. C on the first position in all four nucleotides, G C on the second position in all four codon, and the UCAG in the sequence. Proline. C first position, A second position, UCAG on the third position. Histidine and glutamine are produced. C on the first position, G on the second position, UCAG arginine code. Similarly, like this, all particularly combination has made and different amino acids which are coded by that particular codon is written here. You should remember this AUG which is code for the methionine. And this others also needed to be remembered. Now, here what this short form would indicate, we remember. PH stands for phenylalanine. Okay, then LEU code for the leucine. SER for serine. Then TYR for tyrosine. Then CYS for cysteine. LEU for the leucine that we have already written. Okay. Now then PRO, that stands for proline. Then HIS, that stands for histidine. GLN stand for glutamine, and ARG stands for arginine. ILE stands for isoleucine. MET stands for methionine. Then THR stands for threonine. ASN stands for asparagine. Okay, LYS for lysine. Then SER serine, ARG arginine that we have written already. Then VAL that stands for valine. ALA that stands for alanine amino acid. ASP stands for aspartate amino acid, and GLU stands for glutamine amino acid. Then GLY stands for glycine amino acid. Okay, these all 20 amino acids are there here, which are coded by this particular symbols. Now, by reading this checkerboard, our 64 codon you are there, they are coding for the 20 amino acid. At some position, you will see that 4 or 6 codon are coding for the 1 amino acid. But out of this, you should remember the AG is a starting codon, methionine codon, and 3 are the stop codon. 3 are not coding for any codon. They are not coding for any amino acid, so they are a stop codon. Okay. So this thing happened in particular this. Now after this, we are going to talk about the silent features of the this genetic code. And this silent features, if we talk, dear students, after studying this particular whole checkerboard, first is that the codon is triplet, means codon is made up of 3 nucleotide. And if each codon is made up of 3 adjacent nitrogenous bases, here 61 codon code for amino acid, and 3 codon do not code for any amino acid. They are stop codon. And they are also called nonsense codon because they are not coding for any amino acid, and when they will encountered in the mRNA while reading mRNA, if when mRNA is read by ribosome, if they encounter the ribosome encounter this stop codon in the RNA, the translation will not continue further and it will be terminated without adding the amino acid. Second is one codon code for only one amino acid, that is a specificity. Means UUU code for phenylalanine means it is coding for phenylalanine only. It cannot for glycine. It cannot code for the tyrosine or threonine. It is going to be code for only phenylalanine. So means one codon code for only one amino acid. Hence it is known as unambiguous or specific codon. Now some amino acids are coded by more than one codon. Some amino acids are coded C in reverse. Each codon is specific, means codon will code for only one amino acid. That is not going to be code for 2 amino acids. But there can be a several codon for only one amino acid. Several different type of codon can be possible for the one amino acid. So some amino acid are coded by more than one codon. Hence this code is degenerate as well. Okay. And the codon is read in an RNA in a contiguous fashion. There are no punctuation. Punctuation are not there in particular this RNA. Now, here codon is unambiguous or specific. Exception is GUG. It is ambiguous codon. Why it is ambiguous codon? Because it code for valine and methionine. It code for valine and methionine. It is going to code for this both amino acid, valine and methionine. So that is GUG, which is going to code for the valine and methionine. When the GUG is present in between, then it will code for the valine. But when it is in the initiating position or first position with the mRNA, it is going to code for the methionine. Okay. There is no punctuation, there is no comma, there is read in the contiguous form, but these two you should remember. These two are the important properties. Okay. One is one codon code for only one amino acid, means it is specificity of the codon. But amino acid are degenerated because one amino acid are coded by one amino acid getting coded by more than one codon. Okay. Now the code is nearly universal. Code is nearly universal. Why it is universal? Because for example, from bacteria to human, if you find UUU is going to code for phenylalanine. Some exception to this rule have been found in mitochondrial codon and in some protozoans. In some protozoans and mitochondrial codon, there is a differentiation. But usually, all the codon which code for the particular amino acid, it is similar in all particular different type of organism. Now, AUG, if we talk, has a dual function. It code for methionine as well as it act as a start codon or initiate codon, means from where the translation initiate. Okay. Now, apart from this, there are some exception. Okay. Initiation codon, stop codon, we have discussed. Okay. Some more properties are there. And the fifth property is non overlapping codon. Non overlapping codon means each codon is independent and one codon does not overlap the other codon. Now what is this means? Suppose AUZ is there, GGG is there, and CCC is there. Then this is going to code for first amino acid. This three codon will going to code for second amino acid, and this three going to code for third amino. It is not like that AUG will code for amino acid, one amino, then UGG will code for another amino acid. It is not going to show like overlapping like this, but non-overlapping nature is seen here. And in this, we are going to talk about the some amino acid which are exception to the degeneracy of code. Exception to degeneracy of codon. And such exception to degeneracy of codon, if we talk, then AUG code for methionine and UGG code for tryptophan. And these both are codon. These both codon are known as non-degenerate codon. Okay, so this is the particularly thing which is seen. And one more comma-less nature, because it is read in a without punctuation. So this all are the properties or silent features of the particularly genetic code. So if we want to understand these properties of genetic code in more detail, we can understand by solving some questions. So let's solve one question. If mRNA sequence is if mRNA sequence is this, then polypeptide of how many amino acid get coded by this mRNA. If this is the mRNA polypeptide of how many amino acid is going to be coded by this. If this is the mRNA, if you want to solve this question, dear students, be remember that AUG is just start codon. So first amino acid will be added from starting from here. So it is going to add a first amino acid. This is going to add second amino acid. This is going to add a third amino acid. This is going to add a fourth amino acid. And UAA is the stop codon. So here the termination of the translation will occur and the amino acid are not going to add it. So this 1, 2, 3, 4 amino acid will be present in polypeptide. Okay, so this is the particularly we can say the mRNA sequence. Now after this, here we can say we have understood the particular this question, but here overlapping is not that. If suppose I give a sequence and say that overlapping is present, if overlapping is present, then how many amino acid polypeptide will be synthesized from following mRNA. Suppose mRNA sequence is CUC, CCUACG, CAU, AAC, GGG, and this three dash. Now overlapping is present. Means if we start from here, this is going to add a first amino acid. But then UGC will going to add a second amino acid. Then GCC is going to add a third amino acid. Then this C Cu is going to add a fourth amino acid. Then CUA is going to add a fifth amino acid. Then UAC is going to add a sixth amino acid. Then ACG is going to add a seventh amino acid. Then CGC is going to add a eighth amino acid. Then CGCA is going to add a ninth amino acid. Then CAU, 10th amino acid. Then AUA, 11th amino acid. But when UAA will come, it is a stop codon. So it will be terminated here. Till now, how many 11 amino acid are added? So if your lapping is present, 11 amino acid are added. Okay. So now how this 11 amino acid are getting added, that we have understood. But if you count easily, you can start counting from this will form a one amino acid, then this will code for another amino acid, this will code for third amino acid, this will code for, because they are going to form a successive three nucleotide going to form one codon. So for this, we can count this, for this we can count this, for third amino acid we can count G. So 1 2 3 4 5 6 7 8 9 10 and 11, but UAA will be stop coded, so that will be not coded. So 11 amino acid are added in a polypeptide. So such question can be asked from this particular topic, you should be able to answer. So based on George Gamow, code should be made up of three nucleotides, okay, that we have already studied. Homopolymer are obviously they are made up of same type of nitrogenous bases. So UU is the homopolymer which code for the phenylalanine. Copolymer consists of two different type of nitrogenous bases. Then UUA, GGG, CCC are the homopolymer, whereas GGA that is made up of two different type of nitrogenous bases. So it is a copolymer. These are homopolymer, and this is the copolymer. So fourth is going to be the answer. Now, last we are going to topic the understand a mutation. Actually, mutation we have discussed already in the previous chapter, but today mutation and genetic mutation we are going to refer with respect to genetic code, and whatever we are going to understand this content, that is not more in detail, because all detail part we have discussed in the mutation topic in a previous principles of inheritance chapter. But today just we are going to talk about the whatever given in the NCRT. So mutation and genetic code, if we talk, the relationship between gene and DNA are best understood by the mutation studies. Obviously, they are best understood by the mutation studies. An effect of large deletion and rearrangement in a segment of DNA or in a chromosome are easier to comprehend. So easily, if a deletion or rearrangement any occur in any segment of DNA or in chromosome, that can be easily comprehended by its change in phenotype. Okay, so it may result in a loss of gene or a function. Means if a deletion or a loss in chromosome or anything occur in the particular gene, then there will be a loss of a phenotype or a function. A classical example of the point mutation is a change in a single base pair in the gene for a beta globin chain in human globin that result in the change in amino acid residue glutamate to valine. So in hemoglobin in the beta sheet, if a glutamate is replaced by the valine, that is a particular mutation which is known as substitution mutation, and due to which the sickle cell anemia happens. So due to this mutation we actually occur in the DNA, but due to this the amino acid is getting changed in the hemoglobin, and that lead to the sickle cell anemia, and it result in disease condition called as sickle cell anemia. How change in single nucleotide in a codon alter the amino acid in the polypeptide chain, that is need to be understood easily. So UGC, if single mutation, single change in nucleotide occur, that can be changed into Z. So UGC, it can be converted into the GGU. Okay, okay, UGC. So if we first change U and last we change a C, then GGU can be there. If we keep a G constant, then CGU can be there. If we make a G constant, then AGU will be there. If we make a U constant, if we make a U constant, then G and C replace, then UU will be there. UCU can be there, UAU can be there. If we make a U constant, then UGG will be there. Same way, if you make a UNG constant, then A will be changed, UNG constant, C will be changed, such that a particular change in a single mutation, single nucleotide is affected, like this codon get changed. And you know that cysteine, which when they're particularly converted into this different amino acid, they are going to code for the different amino acid. Means in we can say that change occur in a single nucleotide, and due to this codon getting changed, and when codon get changed, the amino acid it is coding, that is also getting changed. Okay, you can see here. So effects of point mutation that insert or delete a base in a structural gene can be better understood by the following simple example. Okay, point mutation. What will be the point effects of point mutation that insert or delete a base in a structural gene can be understood by the following example. Consider a statement that is made of the following word, each having the three letters like a genetic code, that Ram has red cap. Now, if we insert a letter B in between has and red and rearrange the statement, it would be as follow: Ram has BDCAP. Means whole meaning of this statement has changed by just inserting a B between the has and red. Now, similarly, if we now insert two letters in the same place, say BI would read, then Ram has Bir EDC because further words getting changed because we are adding B I. So now we insert three letters together. Say big. The statement would be read as Ram has big red cap. Now red cap is getting the why because we are inserting a three nucleotide newly. So new amino acid will be added in the if we make a such mutation in the DNA. The amino acid before that and after that will be remain same. Okay. So the same exercise can be repeated by deleting the letters R, E, and D one by one and rearranging the statement to make a triplet word. So this will change the whole Ram has EDC cap because we have removed R. If we remove E, then it will be Ram has DCA cap, DCAP. Then if we remove only D, then Ram has cap. Okay. So why? Because we have removed D. Okay. We have removed red hole. Okay. So like this, the rearrangement can occur, and this conclusion from the above exercise is obvious. So now insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. From where the reading frame is going to be changed? From the insertion or deletion point. So if we suppose this is the sequence CTGAGT, if CTGAGT is there, if we are adding a T, then this is the insertion. If CTGAGT is there and CTG GT is there, we are deleting this A, then means a deletion is occurring. So like this insertion or deletion can occur. So insertion or deletion of three or its multiple bases, insert or delete of one or multiple codon, hence one or multiple amino acid, and a reading frame remains unaltered from that point. Means before and after the reading frame will be remain altered, but one new amino acid will be added if the three nucleotide or a codon with the three nucleotide are removed or added at a same point. So such mutation are referred as frameshift mutation. If we add a one nucleotide or a two nucleotide and if it changes the frame uh, in front of that or after that insertion point, then it is known as frameshift mutation. And we have discussed already this mutation in the previous chapter that mutation are referred as a frameshift insertion or deletion mutation depending on the change. So this forms the genetic basis of proof that codon is triplet and is read in a contiguous manner. Okay, this is the point. This is the particularly point mutation. See, here this is the normal. Here missense means particularly whole genotype phenotype is going to be changed. Here frameshift mutation, this is the frameshift deletion, this is the insertion, and this is the nonsense. Nonsense means it converts into the stop codon. Normal codon when converted into the stop codon, it is known as nonsense mutation. All this mutation we have discussed already in the particularly what we can say in the previous chapter. About tRNA, all thing we have already covered. That tRNA structure also we have discussed in the previous different types of RNA when we have discussed. We have already covered this tRNA in detail. Okay, dear students. So now here today we have already discussed about the what a genetic code in detail. So there are no tRNA for there is a no tRNA for the stop codon. That is a UAA, UAG, and UGA. The secondary structure of tRNA looks like, obviously we have discussed that secondary structure look like clover leaf and 3D structure look like inverted L shape. Okay, so I hope you understood this genetic code. Thank you. Properties, you should remember properties are important point.